I'm not seeing the little arrow by my screen share to approve. Do you have that?
To approve which?
Individual screen sharing?
Oh, I can promote to panelist. Is that what you want me to do?
No, that's what I'm doing now. If that works in terms of screen sharing, then we're good.
Oh yeah, and then I think if we do here, I just changed it to all panelists can share. Who can start sharing? Someone else share.
Okay.
Okay. So we're still waiting for Nelly, is that right? And we're still missing Nelly. Is that right?
Yeah, you can go ahead and start though, and I'll promote her when she gets in.
Sounds good. Okay, everybody. Well, welcome to the Substance of Us session at the Undergraduate Research Symposium for 2020. I'm Professor Josh Snodgrass. I'm professor in anthropology and also the Associate Vice Provost for Undergraduate Research and Distinguished Scholarships. And I'm serving as one of the two moderators in this session. And the other moderator is Alicia DeLouize, who will introduce herself now.
Hi, I'm Alicia DeLouize. I am a graduate student in the Department of Anthropology, and I specialize in biological anthropology.
So the format of our session is it's a 90-minute oral presentation session, and we plan for each presenter, and there are 5 of them, to present for about 10 to 12 minutes. And then we'll follow that by about 3 to 5 minutes of question and answer. And then at the very end of this, after all 5 of the presenters have gone, we'll have a question and answer and discussion session at the end. So the plan for the Zoom session here is it's being recorded. We also want to make sure that people keep themselves muted as much as possible. And then we will take questions as we go. And the moderator, actually Alicia in this case, will be the one who recognizes people, keeps an eye out for people who want to ask questions. So actually, why don't we start out by having each of the 5 speakers just give a little bit of an intro to who they are, and then we'll start through with Alina as the first of the presenters.
Do we just want to go in order?
Sure, that sounds great.
Hi, I'm Alina Salagean, and I'm an undergraduate. I'm a junior and a biology major.
Hi, I'm Cailan Feingold. I'm also an undergraduate. I'm a junior, and I'm also a biology major.
I'm Jeanette Helgerson. I'm a junior undergraduate, and I am a human physiology major.
My name is Dan Tudorica. I'm a biochemistry major and a senior.
And we're still waiting for one more who I don't see here yet, but she will join us and hopefully give the 4th of our presentations. So why don't we dive right in with Alina being the first presenter. She will share her screen so we can see slides, and she'll be giving a talk called Defining the Roles of Conserved DNA Repair Complexes in Maintenance of C. elegans Meiotic Genome Integrity. Take it away.
Thank you. Okay. Okay, so hello everyone. My name is Alina Salagean, and I'm an undergraduate in Dr. Diana Labuda's lab, and my research pertains to defining the roles of conserved DNA repair complexes in the maintenance of C. elegans meiotic genome integrity. So meiosis is the specialized process that many sexually reproducing organisms utilize to create sex cells such as sperm and eggs. Meiosis starts with diploid cells, which divide to form haploid gametes. And then when these gametes fuse together during fertilization, they create a diploid progeny. So during meiosis, double-strand DNA breaks are induced across the genome as a severe form of DNA damage to chromosomes. Meiotic cells undergoing meiosis have 4 copies of the genome. There are 2 homologous chromosomes and 2 sister chromatids. When DSBs are induced, a subset must be repaired through interhomolog crossovers, which are crucial for promoting accurate chromosome segregation. Meiotic cells can also utilize accurate alternative repair pathways and error-prone repair pathways in order to repair remaining DSBs. And although all 3 repair pathways are available to meiotic cells, research has shown that meiotic cells prefer accurate repair pathways as they prevent mutations from occurring. Any unresolved double-strand breaks can lead to meiotic errors, which can sometimes be fatal. So the failure to repair genetic lesions during meiosis can result in the loss of genome integrity, as well as the development of various serious diseases such as infertility, cancer, and birth defects. And so therefore, my research question asks, how do DNA repair complexes interact to regulate meiotic DNA repair? So I began my research by investigating 3 conserved DNA repair complexes: the structural maintenance of chromosomes 5-6 complex, its subunit NSE2, and the BRCA1 BARD1 heterodimer. And previous research has shown that these 3 protein complexes interact to promote DNA repair, but their specific interaction pathway remains unknown. So when we look at each of these 3 repair proteins individually, we see that SMC56 is involved in chromosome structure, and it's also involved in repairing DSBs. NSE2 is known to modify other proteins to influence their function, and it's known to physically interact with SMC56. And finally, the BRCA1 BARD1 heterodimer is commonly mutated in mammalian breast cancer, and it's known to interact with SMC56 genetically. Cenorhabditis elegans is a powerful model organism for studying meiosis, and C. elegans is especially powerful for studying DNA repair. And this is because the hermaphrodite gonad is spatially and temporally organized in a pipeline of meiosis. The oocyte progression rate through the germline is known, which enables a reverse time course analysis of meiotic prophase 1. And we can do this by analyzing progeny of a parent hermaphrodite that's been exposed to a discrete treatment. So due to the pipeline nature of the C. elegans gonad, we can broadly divide the germline into early to mid prophase 1 and late prophase 1, which are the stages at which DSBs are repaired. And the reason why we make this division is because different repair pathways are available at each one of these different windows. So in early to mid prophase 1, meiotic cells can utilize interhomolog crossovers as well as alternative accurate DNA repair pathways. And in late prophase 1, only accurate alternative repair is available. However, at both of these stages of meiotic prophase 1, error-prone alternative repair pathways are suppressed. So when I began my research, my goal was to construct an interaction pathway between SMC56, NSE2, and BRCA1 so as to better understand how they interact genetically to promote DNA repair. I began by creating single mutants for SMC5, NSE2, and BRCA1. And here on the screen, a grayed out protein signifies a loss of that protein. And then utilizing CRISPR-Cas9 genome editing, as well as strain construction methods, I was able to use my single mutants in order to create double mutants for each of these DNA repair proteins. So after creating my mutants, I wanted to induce exogenous DNA damage in my mutants in order to assess whether or not they could repair their genome. And to do this, I utilized ionizing radiation as a form of exogenous DNA damage. So as is shown here, meiotic cells undergoing meiosis in the germline have the DNA stained in blue and DSBs are stained in green. And in a wild type worm that has not been exposed to any source of radiation, few DSBs are observed. However, when worms are exposed to a source of ionizing radiation, we see a drastic increase in the number of DNA breaks, which is indicated by the appearance of the green markings on the DNA. So in order to conduct my experiment, I began by exposing a juvenile hermaphrodite to 2,500 rads of ionizing radiation. And to put this into perspective, a dose of about 1,000 rads is invariably fatal in humans. So next, I singled these worms onto individual plates, and they were transferred every 24 hours for a total of 72 hours at 20 degrees Celsius. And then a brood size was scored. And due to the pipeline nature of the C. elegans gonad, the day 2 and 3 plates correspond to progeny that were in early to mid prophase 1 at the time that the parent hermaphrodite was exposed to the irradiation treatment. And the day 1 plate corresponds to progeny that were in late prophase 1. So after conducting my experiment, I obtained the following data, which is divided into early to mid prophase 1 and late prophase 1. So the data here is for early to mid meiotic prophase 1. On the x-axis are the genotypes of the worms that were assayed, and on the y-axis is the brood viability, which is a metric that we use, which just quantifies the proportion of the progeny that were able to survive embryonic development. So after I obtain my results, I conclude that SMC56 and SC2 interact to promote DNA through a linear interaction pathway in early to mid prophase 1. And I'd now like to take the time to just walk through the data points that were crucial to us coming to this conclusion. So when we begin by looking at the single mutants, we see that SMC56, NSC2, and BRCA1 are all involved in a response to exogenous DNA damage, which is indicated by their decreased brood viability as compared to the wild-type strain. We also see that a loss of SMC5 results in the greatest decrease in brood viability. When we look at the double mutants, and specifically the double mutant for BRCA1 and NSC2, we see that it looks very similar to NSC2 alone. And this tells us that NSC2 and BRCA1 are acting downstream of SMC56 to promote DNA repair, and NSC2 is acting upstream of BRCA1 in order to promote DNA repair. And finally, when we look at the double mutants for SMC56, we see that they look similar to SMC5 alone, which indicates that SMC56 is acting upstream of both NSC2 and BRCA1 to promote DNA repair. So therefore, we can conclude that in early to mid prophase 1, DNA repair is regulated through a linear interaction pathway between SMC56, NSC2, and BRCA1. However, when we look at the results for late prophase 1, we see drastically different results. Based on this data, which is once again presented with the genotypes on the x-axis and the brood viability on the y-axis, we conclude that SMC56, NSC2, and BRCA1 are acting to promote DNA repair through a diverging pathway in late prophase 1. And so when we look at the single mutants again, we once again see that SMC56, NSC2, and BRCA1 are all involved in a response to exogenous DNA damage. And also, once again, we see that a loss of SMC5 results in the greatest decrease in brood viability. However, now when we look at a double mutant for BRCA1 and NSC2, it looks similar to SMC5 alone. And so this tells us that SMC56 is acting as a central regulator, and it can utilize an NSC2 or a BRCA1 dependent pathway in order to promote DNA repair. However, when we look at the double mutants for SMC5, we observe a very interesting rescue phenotype, which looks very similar to the brood viability for the wild-type strain. And so while this initially appears to contradict our diverging pathway, we hypothesize that this may be occurring because SMC56 and NSC2 or SMC56 and BRCA1 may be acting redundantly to suppress mutagenic DNA repair pathways in late prophase 1. And so therefore we can conclude that in late prophase 1, DNA repair is regulated through a diverging interaction pathway between SMC56, NSC2, and BRCA1, and that there may be a potential suppression of mutagenic DNA repair pathways occurring as well. So just to summarize my findings, my first finding is that SMC56, BRCA1, and NSC2 are differentially engaged within meiotic prophase 1 in order to resolve exogenous DNA damage. And so in early to mid prophase 1, DNA repair is regulated through a linear interaction pathway. And in late prophase 1, it is regulated through a diverging interaction pathway. And additionally, we find that there may be some potential redundant suppression of mutagenic repair pathways by SMC56 and BRCA1, or SMC56 and NSC2 occurring in late prophase as well. So for my future directions, I first want to define the specific DSB repair pathways that are regulated by SMC56, NSC2, and BRCA1, and I can do this by utilizing a sister chromatid repair assay developed by the Labuda lab. And additionally, I can utilize single and double mutants for known mutagenic repair pathways. And then I'd also like to determine the cytological dynamics of DNA repair, which I can do by utilizing immunofluorescence imaging techniques in order to visualize repair complex localization. And this can be done by using SMC5, SMC6, and NSE2 antibodies. So finally, I'd just like to say a special thank you to everyone in the Labuda lab for all of their help and support, and especially my amazing PI, Diana Labuda, and my incredible mentor, Eric Torreson. And I'd also like to thank all of my funding sources for making this research possible, as well as the Undergraduate Research Symposium for this incredible opportunity to present my research. And I'd like to open it up to questions at this time. Thank you.
We definitely have time for a few questions.
You guys can submit those via the chat function.
So I'm going to ask a question just as we wait for others to think about theirs, but just I want to know what got you into this particular work that you've been doing.
Yeah, thank you. That's a really good question. I think I've always really loved meiosis and DNA repair in general. And I also really enjoy sort of fertility research. I worked at a mother's milk bank for a while, and so I'm really passionate about reproductive care and reproductive research. And so when I learned that Dr. Diana Labuda had a position open in her lab, I was really excited to apply and to get into this project.
Cool. And speaking of Dr. Labuda, we seem to have a question. Do you think any other DNA repair pathway components are interacting with these different complexes?
Oh, that's a really good question. And I don't think it's one that I had thought of previously. So I'd have to go back and look through my work. In terms of, could you repeat the last bit of the question? Other DNA repair complexes, or was it just DNA repair complexes? I'm sure that there are others, just because we know that, you know, interaction pathways can consist of many different factors. But what those factors could be, I do not know yet, but I would love to spend more time and find out, to sort of further define the pathways themselves.
Great, thank you. Well, we should probably move on. Well, actually, we've got one more question. How about we take one more and then we'll move on. So how did you get the immunofluorescent images of the DNA? And were these images of full C. elegans?
Oh, okay. So the images that I showed previously, I believe it was just, oh, sorry, this is gonna, these images here. So these images were not taken by me, actually. They're from a member of my lab, although I have images very similar to these as well. But these are immunofluorescence staining. So the DNA is stained and in blue, and then the DSPs are stained with an antibody. It's an antibody and then a secondary antibody with a fluorescent molecule, and it stains a recombinase Rad51, which is involved in it marks sites of DNA damage. So that's how we do this, and then we just use fluorescent microscopy in order to obtain these images. But they're taken as tiles, and then we stitch them together as whole gonad images. So this is just a section of prophase 1, essentially.
Cool, thank you. And hopefully we'll have time, maybe the last 10 or 15 minutes of this session, where we can come back to other questions. So, but in the interest of time, let's move on to our next presentation, which is Cailan Feingold with a talk, Development of a New Live Imaging Technique to Uncover the Mechanisms of Heat-Induced Male Infertility.
Perfect. Okay. Hi, my name is Cailan Feingold. I am a member of Dr. Diana Labuda's lab here on campus, and today I'm going to be talking to you about my project in developing a new live imaging technology. Live imaging is an extremely useful tool for researchers. Here in this video produced by the Bowerman lab on campus, we see the early cell divisions of an embryo, Here the DNA is shown in red, and it's being segregated into new cells by the proteins shown in green. And as you can see, this is just about the closest look that we can get to biological processes in their most natural state as researchers. So live imaging allows for us to establish an order of event with the processes that we are studying. It also allows us to answer a number of questions, including questions we may have not had originally in mind. One process I would like to study using live imaging is meiosis. Meiosis includes spermatogenesis and oogenesis. Spermatogenesis, which occurs in males, is the production of sperm, the smallest cells in the body. And oogenesis, which occurs in females, is the production of eggs, which are the largest cells in the body. When spermatogenesis when sperm and eggs come together, they produce a fertilized ovum, also known as a zygote, which will eventually develop into young offspring. There are important differences between spermatogenesis and oogenesis, but before I get into some of the unique aspects of spermatogenesis, there are some steps of meiosis that are useful to keep in mind. First, the pairing of homologous chromosomes must occur. And then the synaptonemal complex, which is seen as the yellow ladder-like structure between the homologous chromosomes, forms. This step is known as synapsis. This allows for the formation of double-stranded DNA breaks, which enables crossing over. These steps help ensure the proper segregation of chromosomes during meiosis. So what happens when the synaptonemal complex fails to form? Well, double-strand break formation is reduced, which means that crossovers are abolished, and this can cause the missegregation of chromosomes. Caenorhabditis elegans is a powerful model for studying meiosis. They are small, transparent soil nematodes, and their transparency makes them ideal for imaging purposes because they can be imaged while they are both alive and intact. They have two sexual forms, the hermaphrodite, which produces both egg and sperm, making it self-fertilizing, and the male, which produces only sperm. While the hermaphrodite is self-fertilizing, in order to have male progeny, we must maintain our worms in mating stalks. Their germlines are also arranged in a spatial temporal gradient, which is useful for studying meiosis because it allows us to track the development of spermatocytes and oocytes as they progress down the germline. As you can see, they have a lot of nuclei, which allows them to produce large amounts of progeny, which enables us to quickly assess the consequences of meiotic mutants. In C. elegans, we have been able to observe some of the same unique aspects of spermatogenesis that we observe in mammals. One of these is that spermatogenesis is uniquely sensitive to temperature changes. Spermatogenesis must occur 2 to 7 degrees below core body temperature, making it the only process that must occur outside of the body. Exposure to elevated temperatures is linked to male infertility and increased risks of testicular cancer. Infertility affects around 1 in 6 couples worldwide, and about half of these cases are due to male factors. Despite its prevalence, though, many of the mechanisms behind male infertility are largely unclear. And so I want to know, what are the mechanisms behind heat-induced male infertility? We know that there are multiple meiotic defects that are occurring in spermatocytes only following heat exposure. One of these is an increase in DNA damage. In my images here, you can see the DNA stained in blue, and DNA damage is marked by green foci. In the no heat shock nucleus, we don't see any green foci indicating areas of DNA damage. However, in the heat shock nucleus, we see a robust increase in DNA damage. This consequence of heat exposure is unique to spermatocytes. We also know that the synaptonemal complex, which if you will recall is involved in synapsis during meiosis, prematurely disassembles as a result of heat stress. Here in these images to the right, CYP1, a component of the synaptonemal complex, is stained in red, and DNA is again stained in blue. As you can see in the no heat shock nucleus, CYP1 forms a cage-like structure around the nucleus, with the lines being well-defined. Whereas in the heat shock nucleus, CYP1 staining is missing in many of the regions of the nucleus, and it appears spotty. This is indicating a potential premature breakdown of the synaptonemal complex. So my primary research question is, what is the relationship between heat-induced DNA damage and synaptonemal complex breakdown? To answer this question, I will be employing the powerful tool of live imaging. One problem with live imaging, though, is that current immobilization techniques fail in male worms. As you can see in this image, the male worm is much smaller than its hermaphrodite partner. Therefore, the physical tools typically used to restrain hermaphrodites fail in the smaller males. So what are our other immobilization options? Well, cold shock is commonly used. However, this would interfere with the heat stress aspect of this experiment. Anesthetics are also commonly used. However, for this imaging protocol, the worms must be immobilized for 3 hours, 2 hours for heat stress and 1 hour for recovery. Therefore, the large amount of anesthesia that would be required would be risky in terms of whether or not the worms would survive the imaging process. So how can we effectively immobilize males while ensuring their survival for the entire imaging process? This is when we turned to a genetic approach, and we identified mutants of UNC genes, which is short for uncoordination, that causes a severe paralysis. We identified 3 candidate genes of focus. We chose these genes based on their location in the genome because they do not lie close to any other meiotic genes, and based on their function because their functions are not known to be involved in meiosis. UNC18 and UNC104 are involved in neuron-to-neuron signaling. Therefore, when they are lost, neuron signaling is prevented. UNC52, which is involved in the extracellular matrix, is involved in cell-to-cell contact in muscles. Therefore, when it is lost, muscle or normal muscle functioning is prevented. However, there's still one problem with paralysis in a male-focused experiment. This is because, as I mentioned before, in order to have male progeny, we must keep our worms in mating stalks. And in order to mate, worms must be mobile. Therefore, the paralyzed UNC worms would be unable to produce male progeny because they would be unable to mate. So how can we use these genes for immobilization while still allowing for mating? This is when we turned to the auxin-inducible Degron system, which is a system adapted from plants to work in worms. It allows for the conditional degradation of target proteins in response to the hormone auxin. It requires 2 transgenic components, the protein of interest with a Degron tag, and the transport inhibitor response 1 protein, also known as Tier 1. Upon exposure to auxin, Tier 1 is activated to degrade the Degron-tagged UNC protein, allowing for the conditional paralysis. This system is ideal because upon removal of auxin, the Degron-tagged protein can be remade, allowing for us to reverse paralysis. Therefore, with this system, we can maintain our worms in mating stalks and paralyze them when it comes time to image. Before we tagged these proteins, though, we wanted to ensure that these mutants did not interfere with meiosis or fertility, so we performed fertility assays. To look at brood size, we looked at our average brood size per worm per day. In this graph on the left, we see average brood size on the y-axis, and on the x-axis is the day that the progeny number was recorded. As you can see, the wild-type worm is laying much more progeny than the mutants, especially on day 2. However, the mutants are grouping together and showing similar numbers. Their laying of less progeny may be indicative of a slower progression through the germline, which we will need to keep in mind as we proceed. We also wanted to ensure that meiosis was not being interfered with in these mutants. So we looked at brood viability data, which is the proportion of living progeny to total progeny, which includes both live and dead offspring. Here on the graph to the right, brood viability is shown as a percentage on the y-axis. As you can see, the mutants look very similar to the wild-type data. Therefore, we don't see any obvious meiotic defects in these mutants, and we decided to proceed with them. Currently, we are making the conditional mutants, and once they are made, I will again perform fertility assays on them to ensure that still meiosis is not being interfered with. I would also like to answer my primary research question in defining the relationship between heat-induced DNA damage and synaptonemal complex breakdown. For this, I can use my live imaging experiment. I would also like to determine an order of events during heat shock and recovery. For example, being able to answer questions like how long into heat shock before DNA damage begins to occur, or what does the recovery process look like? For this, I can also use my live imaging experiment. I would like to thank all of the members of the Labuda Lab, especially my principal investigator, Dr. Diana Labuda, and my mentor, Dr. Corey Cahoon, for all of their mentorship. I would also like to thank the Knight Campus Undergraduate Scholars Program for their support in this project. and to the Undergraduate Research Symposium for the opportunity to present. And now I will open it up to any questions.
Great, thank you. Anybody have a question? I guess while we're waiting for people to type in their question, I will ask one. You did a really nice job laying out this really nice story about the, you know, these questions that you have. And like all science, I think, you know, there's oftentimes a lot behind the story in terms of the technological challenges or the experiments that go wrong. And I just wanted to hear a little bit about maybe something that you experienced that really was a stumbling block or something that was totally unexpected in this. And then how did you get beyond that?
Definitely. I think that not necessarily having to do with this live imaging technology, but in general, joining a research lab, I was not prepared for how kind of complex it is to create like mutant strains and how much time and energy that takes, and sometimes a lot of troubleshooting involved. And so that took a lot of my time over the past few terms when we were actually on campus. But we got that figured out, but unfortunately we're not on campus at the moment to continue with some of this, but I look forward to getting back into it.
So we've got a few questions here. Yes, sounds like a long process. How long did it take to develop the process of using the UNC gene?
So we already have some mutants of this gene available in lab that are not conditional. So therefore we were able to perform the fertility assays pretty quickly once we had decided on these mutants. The construction of the conditionally tagged with the dead-ground tag mutants is ongoing. So unclear on the timeline at the moment, but hopefully as soon as we get back, we can continue with them.
Cool. And then I have one other question, and then we'll move on after this. Does the live imaging technique have the potential to alter the observed meiotic process? So is brood size a reliable indication of meiotic viability?
That's a good question, and something we definitely need to keep in mind. I think always being aware of how our experiments can interfere with the process that we're looking at is important. I think more so than the brood viability, the brood viability, not the brood size. The brood viability is a good indication of these mutants being stable in terms of meiosis.
Okay, well, I'm going to ask one more since I cut Hans off, Hans Dreyer, last time. I'm going to let him ask his question here. So is there a circadian component to the HS exposure response?
That's a great question. I don't know the answer to that question, but it's interesting to consider.
Okay, maybe we can come back to that at the end.
Yeah.
I hope people are all able to stick around at the end, and we can discuss some of these other questions coming up. So we are now moving on to our third presentation, Jeanette Helgerson from Human Physiology, presenting differences in old and young patient-derived myotubes response to amino acid stimulation.
Great, thank you. Let me set up the sharing. I believe this should be all good. Do that. All right. So again, I'm Jeanette Helgerson, and the name of this presentation was Differences in Old and Young Patient-Derived Myotubes Response to Amino Acid Stimulation. So just some background. We need to know how we get from a muscle satellite cell, or a muscle stem cell, or in this figure, a myogenic precursor cell, to a muscle fiber, which is what's in the body. So myogenic precursor cells are determined to become myoblasts. These myoblasts proliferate, meaning they produce more of themselves. They then differentiate into myotubes, which are characteristically multinucleated. And that's where we focused on our study for the myotubes. However, in a human, these myotubes would fuse to become muscle fibers. This research was really important because older patients have a hard time gaining and maintaining muscle mass, particularly after surgery. This lack of muscle mass can lead to health issues, impaired daily life, and even death. In a previous study from our lab, when total knee arthroplasty patients received essential amino acid supplementation, these patients had increased satellite cell numbers, which was associated with reduced muscle atrophy when compared to placebo control. Learning how this patient population's muscle cells respond to amino acids may help these patients reduce atrophy and or maintain or even gain muscle. So what amino acids did we want to use for our study? In another previous study from our lab, we found that satellite cell members were shown to increase after 7 days of essential amino acid ingestion. Another group, Sabatini group, found that leucine, arginine, and lysine in combination showed the greatest activation of the mTORC1 pathway, which is an anabolic pathway indicating that cells are growing. So to the right, you can see a Western blot image where in the top bar, a darker band indicates more proteins, which indicates the cell is in a growth state. So with that 20, meaning 20 essential amino acids, there's a activation of this mTORC1 pathway. And we don't see that same level of anabolic stimuli until the LRK group, meaning the LRK, or leucine, arginine, and lysine, is a good mixture of amino acids to use for our study. Our goal was to understand the mechanisms that promote growth and proliferation of these human-derived muscle cells and to identify key differences in the response of young and old human-derived myotubes to this anabolic or growth stimuli. We hypothesized that aged cells would exhibit a diminished and delayed signaling response to amino acids as compared to the younger cells. For subject recruitment, we put posters all around the Eugene area, and because we were trying to find older patients, we used this Eugene recreational website to find community centers as well as meal centers focused for seniors so that we could go and tell them about our study. And then the next slide has images of a human biopsy, so please be aware of that. In the human biopsy, the first image shows the instruments used. This biopsy is taken from the vastus lateralis of the quadriceps. Local lidocaine is applied to make sure that it is numb and the patient won't feel anything. A small incision is made and the biopsy needle is inserted. A small piece of the muscle is taken into that needle and taken out. The third image shows what the incision site looks like after the needle has gone in. We then cover that with steri strips and put a tegaderm on it to ensure sterility. That small muscle piece is brought into the biosafety cabinet pictured below, and we cut the tissue into really small pieces, and we place small portions of that in the 12 well labeled on, or pictured on the side, and we leave them in an incubator to grow. The one potential downside of this method is we might get cells growing that are not necessarily muscle cells. So we tried a different cell explant method as well, called digestion. It starts off the same way. The tissue is still cut into small pieces, but we place those into a digestion mixture, which would hopefully release the satellite cells. We then physically filter it to remove the debris and hopefully get a solution that is just the satellite cells, which we place on a flask to grow in the incubator. The image on the right shows what that looks like directly after the physical filtration under a microscope. So at this point, we have satellite cells and we put them in growth media to begin the determination phase to become myoblasts. We then use MAX, or magnetic activated cell sorting, which is a purification process where we use metal beads to tag the cell surface marker CD56, which is a satellite cell marker. We then pass that through a magnet, pictured on the right, to put the tagged cells, attach them to the magnet, and all other types of cells will hopefully get flushed out in order to further purify our sample. At this point, we let them grow into myoblasts, and we're in the proliferation phase. And once our cells reach 80 to 100% confluency, meaning they cover 80 to 100% of the bottom of the well, we take a small sample and we count them on the hemocytometer pictured on the right. This allows us to split cells into flasks with approximately 50,000 cells per flask, which allows us to control between subjects. And then we take another small subset and we type them to ensure that we did purify the sample enough. The first image is stained for DAPI, which is a marker of cell nuclei, and it allows us to count the total number of cells in these flasks. Then we also stain it with TE7, and TE7 is a marker of fibroblasts, which is the most common other type of cell that we get. We consider a sample pure enough if we have less than 20% of TE7 stained cells, which this sample did have. At this point, we put them in differentiation media to allow them to become myotubes. And we begin cell lysing, which is a procedure that kills the cells, breaks them open, and creates a protein homogenate called a cell lysate. This cell lysate is quantified and equalized between subjects. We load them onto an 18-well gel, and we separate them by size using electrophoresis. We then transfer these proteins from the gel to a PVDF membrane. We incubate them with the appropriate antibodies, and we image them using the Bio-Rad molecular imager on the right. We specifically were looking at the proteins AKT, which is a marker of anabolic growth, and GAPDH, which is our housekeeping protein, meaning our experiment should have no effect on it. So if myoblasts are grown in growth media to proliferate and then put in differentiation media to become myotubes, both of these media include growth factors, which are anabolic. So to ensure that in later experiments that amino acids would be all that was acting on the cells, we needed to find out how long they had to be serum starved in PBS to have no activation of the growth signaling pathways. So we tried serum starving for 1, 2, 3, 4, and 5 hours. And because we used AKT as a marker of growth signaling, the time points that had the smallest amount of AKT would be what we used for later experiments. So this brought us to our first learning moment of the summer. GAPDH is a far more prevalent protein than AKT. So because these proteins are different sizes, they're in different places on the membrane. And if we allow GAPDH to go to saturation, meaning we get a dark band here, that means that we didn't get enough signal from the AKT because there's far less AKT. And we figured out that we needed to allow AKT to reach saturation as well. With the preliminary plots that we got, it suggested that cells showed AKT plateauing at 3 hours. However, we were unable to get a representative image for the slide because of COVID-19. And then unfortunately, right at the end of the summer, right after we got this preliminary blot, we had yeast contamination of our incubator due to airborne yeast. We tried to use amphotericin B to kill the yeast. However, it was a little too late and the fragile primary myoblasts became nutrient starved and died, which was really devastating because we had a lot of samples in that incubator. The one positive was it led us to implement even stricter sterilization procedures and preemptively include amphotericin B in all growth media. So through the summer research, we perfected many methods. We found new avenues of subject recruitment, particularly for older populations. We tested digestion as a viable cell explant method. We documented cell growth timelines from satellite cells to myoblasts to myotubes. We gained experience completing the Western blot procedure. We found that serum starvation for 3 hours was enough to diminish anabolic signaling left over from the differentiation media. We increased incubator sterilization procedures, and we gained experience dealing with contamination of study materials. As you can probably tell, the aims for this research project have yet to be completed, so the main future goals are to compile the results to find if/what differences young and old human-derived myotubes have in response to that growth stimuli. Because we found a serum starving time that has been established, amino acids can now be added, and we can know that any growth stimuli we see will be due to the amino acids rather than the incubation media. The results of the study could potentially help people, particularly older subjects, reduce muscle atrophy/increase their muscle mass, which could be particularly beneficial prior to surgery to reduce muscle loss, speed up recovery, and return these patients to a higher quality of life sooner. We wanted to acknowledge that funding was provided by the Peter O'Day Fellowship in Biological Sciences through the Undergraduate Research Opportunity Program. So thank you very much for funding this research. And are there any questions?
Well, thank you. Anybody have questions? And I will start off with where do you see this research going? Not just the particular study, but I guess where do you see it taking you long term?
Like my future, or as in like what this could be applied to in the future?
I guess more you, and sort of whether this is something that you're going to continue on in the future with graduate school, or whether you see, more about you.
Yeah, definitely. I'm particularly interested in going to medical school, but I would love to continue research there. I'm particularly interested in muscle function, which is why I'm very interested in being in a muscle lab. And I would love to continue to find ways to help patients before and after surgery, particularly with that muscle atrophy that we see from surgery. And something like this where we could give them an amino acid supplementation would be very, very cool.
And particularly around aging too, or all age groups?
Particularly aging, just because they tend to have the biggest atrophy that we see, but all patients would be great.
Mm-hmm. And especially all of us sitting around at home all the time with the
yes.
muscle atrophy.
Would be nice.
Okay, we've got a question here. Do you plan to run experiments in your senior year, say on C2, C12 and humans?
Definitely, yes. C2, C12 is the mouse cells, which is kind of the industry standard for this type of experiment. And I would love to get more of that information from C2, C12 that we could then apply to human muscle cells. So I plan on continuing research as long as I can.
Cool. Okay, so we got a couple more questions. With COVID-19 patients being bed rest for weeks, do you think any of this work could be applied to help protect these patients from extensive muscle loss?
Definitely. Because we've had previous studies show that this amino acid supplementation could increase satellite cell numbers, this could potentially help anybody that is having muscle atrophy, especially long-term when they can't get up and exercise to increase that. A supplementation could be very beneficial.
Cool. About one more here before we move on. Do you think the health status or pre-existing health conditions of your subjects, particularly the older patients, may affect the results?
Certainly. So we did try to control for that as much as possible, and we did require that the older patients were healthy and didn't have specific pre-existing conditions. However, there is, of course, some level of just having an older population that will be different from the young population, which is actually what we were trying to look at. So I think that it's good that we have that difference. And because we controlled for the other types of differences, I don't think that it would be that important, if that makes sense.
Perfect. Well, thank you.
Yeah,
and
thank
again,
you.
We'll be able to come back to these questions at the end of the session. So we're going to move on to our fourth speaker today is Nelly Nouboussi from biology. And she is now here and can share her screen. Glomerular signals underlying olfactory navigation.
Hi everyone, my name is Nelly Nouboussi and I'm going to be presenting my thesis, which is titled Glomerular Signals Underlying Olfactory Navigation. So why is it important? So why is it important to study olfaction? Well, the olfactory system is the least covered sensory system in most neuroscience classes, although it plays a very important role in many aspects of our existence. Attraction partially relies on olfaction, as we tend to be attracted to people we perceive as smelling good. Taste also relies on olfaction, which is why when you have the cold, certain food might not taste as good. It also serves a protective function, as a food smell can sometimes be an indication of what condition it is in, preventing us from ingesting bad food. Additionally, impaired olfactory function can sometimes be a marker for depression. Beyond its use to human, the olfactory system is important for many animal species, such as dogs. And studying the olfactory system is important in general because it gives us insight into general brain function. The mouse is the dominant model organism for studying systems neuroscience in general, but in our lab we use it specifically because it has a very developed olfactory system with way more odor and receptor genes than humans. Humans only have about 300, but mice have like 1,000. And also for the longest time, mice were the only animals for which genetic engineering was available. So scientists could design mice to express different indicators in different parts of the brain. So I'm going to go more into some background. In terms of the anatomy of the olfactory system, we know a lot about how odor information is processed. So odor molecule will bind to olfactory sensory neurons in your nose, and these olfactory sensory neurons converge into structures called the glomeruli, which are these here. And in the glomeruli, they make synapses with mitral and tufted cells, and these are the output neurons of the brain. And mitral and tufted cells send information to the olfactory cortex, where it is processed in order for us to respond accordingly. And I am focusing specifically in the glomeruli here with the mitral cells. So my lab and others have studied the behavioral structure of olfactory search, which pretty much means that we have analyzed how mice move in a search task, what kind of sampling movement they use to solve the search task. But our next goal is to be able to compare these sampling movements directly against sensory input. We want to be able to visualize a correlation between what the mouse is doing and how the brain is responding at that time. And the first step in this goal is what my thesis is really about, which is getting the imaging technique to work. So during the time of this thesis work, I've been troubleshooting imaging and surgery techniques necessary to actually begin the experiment. And during this period, we've been doing our experiments on head-fixed mice because it allows us to have better control, although ultimately our goal is to be able to image freely moving mice. But we're starting with head-fixed mouse first because, again, it just eliminates a lot of different confounds. Because this is really a method thesis, there's a lot of different methods I need to talk about. So here we go. So in order to be able to visualize activity in the brain, we injected viral vector encoding fluorescence calcium sensor in the olfactory bulb. And so once these sensors are expressed, they can bind calcium, and upon calcium binding causes a conformational shift that results in an increase in fluorescence beyond the basal level. And we injected 2 different viruses. The first one is called AAV-GChem7F, and this one is non-specific and non-Cre dependent. So it pretty much means that it can be expressed anywhere in the olfactory bulb. The second virus is called AAV-GChem7F flux, and this one is Cre dependent. So what this means is once you inject the virus in the brain, initially the gene is inverted, which means it cannot be transcribed and you cannot express the GChem protein. So you can't really see anything. But if these cells express the Cre recombinase protein, what the Cre recombinase protein does is to invert the gene in order for you to be able to transcribe your gene and express your protein. So we're using this virus to restrict expression of our fluorescence protein in specific region of the brain. Now I'm going to talk about the 3 different mice that we're going to be working with, that we worked with. V6 mice are wild type. So these are the ones that we injected the non-Cre dependent, non-specific virus in, so it can be expressed anywhere in the brain. The second mouse strain is called Tbet-Cre, and these one express the Cre protein only in the mitral and tufted cells. So these mice receive the Cre dependent virus. And again, like I told you earlier, the mitral and tufted cells are the output neurons of the brain, so they're the one that we're really trying to focus on. So by injecting the Cre dependent virus in these mice who only express Cre in the mitral and tufted cells, we can restrict the expression of GChem in that region. And the third mouse strain is called Tbet-Cre/AI148D. And this mouse strain is the result of crossing of Tbet-Cre mouse with AI148D mice. And these mice contain a conditional GChem6F allele with a TRE promoter, a stop cassette flanked by LOXP site, and the GChem6F gene. So this GChem6F gene cannot be expressed because of this stop cassette here. But if you have the Cre protein present, what you can do is cut at the LOXP site here and remove the stop cassette. So this picture is just a generic image to kind of show what's happening. So the EGFP represents your GChem6F gene, and the target gene kind of represent the stop cassette. So the Cre recombinase protein will cut at the LOXP site and remove the stop cassette so we can express the GChem6F. So again, in this case, only the cells that express Cre will be able to express GChem6F gene, the GChem6F gene. And so we decided to use this mouse strain because it pretty much eliminates one of the steps in our procedure, because these mice didn't receive any virus injection because they already encode the gene in their genome. So they have already been engineered to encode the fluorescence gene. So you're pretty much eliminating the virus injection step. We wanted to see if maybe this will give us better expression compared to the Tbet-Cre mouse, the Tbet-Cre mice. In terms of the surgery that will perform, virus injection surgeries pretty much just consist in inserting a needle inside the brain, injecting your substance and removing it. And this picture shows the general area where we're usually inserting the needle. And this one here shows you the different site where we usually insert the needle. So it's closer to the eye. And for this experiment, we were just doing two two injection holes on the left side of the olfactory bulb. We didn't really do any on the right. We also perform cranial window/head bar surgeries, and this one consists in removing a part of the skull and replacing it with a window so you can actually image so you can actually see the brain. And we're also placing a head bar in front of the ears to be able to head fix the mouse during the imaging session. So we also did some histologies, and we looked at the brain using a 2-photon microscope, and we also used a fluorescence microscope to look at some of the histology sections that we got. I'm going to go into the results section now. So one thing I do want to mention is initially we were not really seeing any expression of the, we weren't seeing much expression of the GChem gene, so the GChem protein. So we decided to inject 2 mice with the fluorescence tracer called Dio, because it can allow us to look specifically at the injection region, and it's also very vibrant. So I'm just going to show you those images and kind of what we learned from these sections. So if you focus just on the top layer, we shouldn't really see anything with the CY3 channel because Dio absorbs light at similar frequencies as GFP. So it also reflects green, not cyan like for the CY3 channel. So what these images allow us to see is that there were a lot of issues with the injection setup that we were using. If you look at this image here, usually when we're doing the surgeries, we insert the needle, inject the virus, wait for it to be absorbed, and then pull out about halfway, and then wait some more, and then pull all the way. And what you see here is that some of the virus is being released at the halfway mark, which is not supposed to be happening. And we don't also know what this is. And from the third image, we see that some virus is also being released as we're pulling out the needle near the surface of the brain, which is, again, something that we're not expecting. So these images led us to kind of conclude that we had a lot of problems with our injection needle, injection setup. We couldn't really control we couldn't control how it was working. So we decided to switch from what we had been using, which is called the NanoJect syringe, to the Hamilton syringe. The Hamilton syringe was used by a different lab, and they told us that it's definitely better for control and it's also easier to use. So since then we have been using that, and it's definitely been working a little bit better. Now I'm going to talk about the result from the 3 different mice, three different mouse strains that I talked about earlier. So for the B6 mice, we do see some expression of our virus, which is here and also here, but it's definitely not as much as we were expecting. If you remember, these B6 mice are wild type, so you can express GChem pretty much everywhere. So we expected to see way more expression throughout the brain. And it's possible, it's because it could be for multiple reasons. Maybe we didn't inject enough virus, or because we took these images kind of late, it's possible that some of the virus might have been degraded at this point. So we're going to try and do it differently in the future. But for these mice, we did see some expression, which is not which cannot be said, which we can't say for the other 2 mouse strains. So for the Tbet-Cre mouse, we don't really see anything. So if you remember from what I told you earlier, we're really focusing in the mitral layer, and I tried to outline it here so you see where we were supposed to see fluorescence, but we see more out here, which is definitely not what we're looking for. So that's why we conclude that this is probably just background fluorescence. It's not really what we injected. And because this is the region where you're expressing Cre. And if you remember, these mice receive the virus that is Cre dependent. So you should only see expression in regions where Cre is being expressed. But that's not what we're seeing. And we also confirmed that result with these images because, like I mentioned earlier, we only injected we only drilled 2 holes only on the left side of the bulb, but we're seeing expression in both. We're seeing general expression in both sides, which is not what we're expecting. So this is again confirming the idea that this is just background fluorescence. It's not real. And one of the things that we were speculating on why this is the case is kind of the same reason that I mentioned earlier. I took these images maybe about 3 to 4 months after the injection had been done, which is a pretty long time. So it's possible that at this time all the virus might have been degraded. So we're going to try to do histologies closer to the injection period. Now for the third strain of mice, the result is again the same. We don't really see expression in the mitral layer like we were expecting. We see just this band. We really have no idea what this is, but we definitely know it's not the virus that we injected. So it's again probably just background fluorescence. And so usually you can restrict the expression of Cre by feeding mice with doxycycline, but we didn't feed these mice with any doxycycline. So they had been expressing Cre throughout they had been expressing GChem throughout their whole life. So it's possible that this continuous expression might have resulted in the frying of the bulb. So we don't really know, but we're also going to try to do doxycycline treatment up until before we are going to start the experiment to see if that could give us better result. But these two set of mice strains didn't really give us what we were expecting. So the cranial window surgery is definitely a really hard surgery. I think I performed maybe like 10 and I had maybe 1 or 2 that were successful. So it's a surgery that has a really low success rate. And just there's also a lot of different issues with the microscope that we're using. So we haven't really been able to get a lot of good images during our live session with the 2-photon microscope, but there was 1 or maybe 1 or 2 that were good. So those are the ones that I'm going to present. I was supposed to do more imaging during spring term, but obviously that didn't happen because of the coronavirus. But I'm just going to present what I had before, which was not very much. And so this image was taken with the 2-photon microscope. And so it does allow us to see some cellular structure. So the blue arrows are pointing at the dendrites and axon, but the red arrows are pointing at cell bodies. So the 2-photon definitely allows us to see structures with a little bit of detail, which is why we are going to do the experiment in the future when we're doing live mice with the 2-photon. The histologist was just to confirm that we're actually getting expression. So this is just like one of the very few mice that we got that had a good result. And I also have a video to kind of show this modulation. If you focus on the blue square, you can see that it's like flashing. So you're actually getting GChem expression while the mouse is being exposed to air. And this experiment, it wasn't exposed to any odor, just air, and it's just breathing normally. Right now it was moving, but it's just breathing normally, and you can see that the GChem is actually modulating, which is what we're expecting. So we hope that once the technique is working perfectly, we can actually start our experiment to get what we're looking for. In terms of future direction, like I mentioned earlier, we're going to try to do histologies just a few weeks after injection and the doxycycline treatment right before we start doing the imaging experiment to see if that could give us better expression. And we're also going to keep working on the cranial window surgery because, as I told you guys, it's really hard to actually get a good result. And once the technique is actually working, we're going to be using it to monitor and manipulate activity in the glomeruli during active sensing behavior, which pretty much just means, again, that we're going to try to to visualize a correlation between what the mouse is doing and how the brain is responding. And one of our hypotheses is that olfactory navigation is driven by a comparison of glomerular signals across multiple SNPs, which means that what the mouse decides to do or how the mouse decides to move next depends not just on the previous SNP, but an integration of different signals from multiple SNPs before the current one, if that makes sense. Okay, and I would like to thank my PI, Dr. Matt Smear, for letting me work in his lab, my mentor, Amanda Welsh, and Dr. Brown and Finley for helping me with this project, and also other members of the Smear Lab for their support, and the Neil Lab because they helped me with some of the techniques, and also the Institute of Neuroscience. And finally, the Undergraduate Research Symposium for giving me a platform to present my research. Thank you. And now we can go into questions.
Maybe have time for one question, just in the interest of time. I'd love to be able to have some time at the end for all of us to discuss. Anybody have a question? I'm trying hard not to ask my own questions. Okay, since nobody's jumping in right now, I'm going to ask a quick question, which has probably not got a quick answer. But just thinking about this in terms of relevance to understanding humans, I guess what's the long-term possibilities of this research and this kind of research?
So definitely this research doesn't have as much relevance to humans, but it can be applied in terms of just understanding how our olfactory system works. But like I said, it's more, right now it's really being used with like dog research. I know one of my mentors in my lab is working to like understand how search dog works, like figure out how people are lost or to just find things. So I know that it can be applied to that, but I'm not really sure how we can apply it directly to humans. I'll say it's more relevant to like mice.
Right.
But it gives us insight into like general brain function, so we can make comparisons between how the olfactory system works and how other systems work, like the visual system and other sensory systems. So in that case, you can say that it can be applied to human, but not necessarily directly.
Right.
Yeah.
Awesome. Cool. Thank you. Okay, moving on to our last talk of the day. Dan, I don't see you, but I see your name. So I'm assuming you're there in person.
I'm here and ready to go.
Perfect. And giving the last talk of the day, the role of chemoreceptor zinc-binding domain in bacterial signal transduction.
Okay, great. I'll start sharing and presenting. Great. Okay, so my domain is, my presentation is formally called the oxidation of CZB domains by hypochlorous acid, but really what it's all about is trying to understand how gut colonizing bacteria sense their environment, and really why does this matter for us? And so essentially I'm trying to figure out how do bacteria sense their environments and find safer niches in order to colonize and thrive within their human hosts. And so in trying to answer this question, it's helpful to try and think about what obstacles gut colonizing bacteria might face. So obviously, when you ingest a bacteria and it falls into your stomach, the bacterium in question has to deal with like a great deal of obstacles, not least of which is the very concentrated stomach acid that it immediately finds itself in. In addition to that, the human body has this mechanism where it recruits immune cells to sites of tissue damage and starts pumping out large amounts of bleach, up to 5 millimolar, which is about maybe 1/5 the concentration of household bleach you might use. And in addition to that, when the human body senses that it's being infected, it has various mechanisms to try and hide away key nutrients from the bacteria that's infecting it. And a good example of this is iron, for example. When your immune system kicks in, your body starts to sequester away iron so that bacteria can't take advantage of it and use it to thrive. And so trying to understand how bacteria deal with all of these sort of opposing forces was a big motivating factor. And to kind of illustrate how bacteria tend to deal with this, I'd like to talk about one example, the bacterium Helicobacter pylori. This is a highly infectious bacterium that over 50% of the world's population has. In some areas, it can reach up to 100%. So literally almost every single person in a given area might have this bacterium. And it's responsible for causing ulcers, cancers, and once you get it, it persists for decades in the human body. And so what I was really curious about was trying to understand why this bacterium was so effective at colonizing the human body. How is it so good at staying inside a very hostile environment? And so researchers found out that one of the key strategies it uses is that when it lands within the stomach, it's drawn towards sites of inflammations and weaknesses in the stomach wall where it kind of buries itself and survives long term. And so you can see that on the image down here. The red indicates mouse stomach wall tissue. The green dots indicate the bacterium, and the star indicates a site of inflammation caused by a laser. So you can see over the course of 30 minutes, inducing inflammation really like gathers all these bacteria to the site of inflammation where they start happily residing and colonizing. And so a big consideration was what could the bacteria be sensing that draws them to this safe harbor? And one possible candidate was actually bleach, because as I said before, at sites of inflammation, the human body, or the mouse body in this case, starts pumping out large amounts of bleach in an attempt to disinfect itself. And so work done by my mentor and I identified the protein responsible for bleach chemoattraction. And that data is shown over here. You can see that in the presence of bleach, the bacteria are drawn very strongly. So over here is a pipette that's slowly dispensing bleach, and over here is a pipette that's slowly dispensing buffer. So you can see here that a pipette that dispenses bleach draws a great deal of the bacteria towards it. And so work done by my mentor and I identified that the protein chemoreceptor TLPD was responsible for this bleach chemoattraction. And so this explains really neatly how pylori can deal with the damaging effects of stomach acid and having iron sequestered away from it by sensing the bleach that's pumped out by sites of inflammation and then swimming towards it. And so this is the structure of TLPD, the chemoreceptor that my mentor and I identified as important for bacteria being attracted to it. And over here is what we suspect would be the sensing domain. So while we understood how what the importance of the chemoreceptor is and how it, for example, signals the rest of the cell, what we didn't necessarily understand was what biochemical mechanism it used to sense bleach in the first place. And so that was kind of the main question of my research, was to try and understand how this domain over here, the chemoreceptor zinc-binding domain, interacts with oxidative species and therefore transduces that signal to the rest of the protein and then to the rest of the cell. And so as a short background on chemoreceptor zinc-binding domains, they're a class of sensing domains that are attached to diguanylate cyclases, which are enzymes involved in biofilm formation, essentially whether a bacteria is free swimming or kind of in this gross film stuck to the wall of something. And it's also attached to chemoreceptors, which help define the direction in which bacteria swim. These are present in a very wide variety of bacterial species, and it's also characterized by some very highly conserved high affinity zinc binding sites right in the center. And so because this zinc binding site is very well conserved across all species, it's quite possible that by studying just a single domain, we can gain crucial insight into how all domains of this type work. And so the hope is that we can understand how sensing works for this one domain and therefore understand how these wide classes of bacteria are capable of sensing bleach and reacting towards it. And so my hypothesis was essentially that sensing was performed by this cysteine over here. It's part of this highly conserved zinc binding core. So my hypothesis was that a molecule of hypochlorous acid or bleach would find its way to that cysteine and then oxidize it. And upon oxidation, initially the cysteine would be bound to the zinc core, but oxidation would cause it to release it, resulting in some structural change. And so that was the hypothesis I was going after. And critically, this hypothesis offered me some kind of testable physical metrics I could use to understand how the protein functions. Firstly, if it's true that this cysteine acts as a sensor by oxidation, we should be able to oxidize it and observe it being oxidized. And secondly, we should observe some very regular stereotype structural transition upon oxidation with the bleach molecule. And so that offered kind of two testable metrics that I could go about to try and prove or disprove this hypothesis. And so in order to compare, I used these two point mutants, one where the zinc binding cysteine was mutated into a histidine, that's right here, and one where I mutated the zinc binding cysteine into an aspartate. And so these are essentially the same proteins, the same domains, but I took away that one cysteine that I thought was important for the sensing function, essentially in order to provide a negative control. And so I hypothesized that this H mutant would be sort of a replicate of what the unoxidized state would look like, and this aspartate would be a replicate of what the oxidized state would look like. So this one would always be, for example, in the on state, let's say, and this one would always be in the off state, because histidine binds zinc, but aspartate does not. And of course, these are only hypothetical structures, so this is just what I imagine they would look like because I don't have crystal structures of these exact mutant proteins. And so the first experiment I want to go about was to answer that question of how readily is the zinc binding cysteine oxidized. So essentially what I used is an antibody and chemical adduct system that basically gave me a readout of how much oxidized cysteine was present in each individual sample. And basically the way the raw data looks is this field of black dots basically. And the darker the dots are, the more oxidized cysteine is present in each sample. So essentially what I did is I titrated increasing concentrations of bleach into the wild type CZB domain, a C52H mutant, the one where I replaced that critical cysteine with a histidine, and also the one where I replaced it with an aspartate. So you can see here that the wild type one is oxidized much more readily than the histidine or the aspartate mutant. And so the wild type strain has two cysteines, one that's critical, the one I want to analyze, and then one sort of non-specific cysteine that just happens to be on the surface of the protein. And this non-specific cysteine is also present in the H and the D mutants. So I would expect that the signal from the wild type would at least be twice as strong as the signal from both of the mutants. And what I actually found was an even more stark difference between the wild type and the mutant proteins. So you can see here, these two curves indicate the mutant proteins, and this is the wild type. So at certain portions, the wild type signal is something like 10 or even 20 times as strong as the D mutants. So this indicates a couple of things. Firstly, it indicates that the zinc-binding cysteine is much more reactive to bleach than a generic surface-exposed cysteine. And so this probably indicates some evolutionary purpose in the cysteine being so finely tuned to sense bleach. Furthermore, this cysteine residue was actually specific to bleach over other oxidants as well. So this blue curve over here shows the signal from a titration of bleach, and then this curve down here shows the signal from a titration with hydrogen peroxide, another common biological oxidant. So not only is this residue very, very sensitive to bleach, it's also specifically tuned to sense bleach over other oxidants it might encounter. And so to answer the second part, I wanted to get some information about how the structure changes post-oxidation. And to do that, I used a method called method called tryptophan fluorescence. So essentially what tryptophan fluorescence does is it reports on the solvent environment of tryptophan residues. Basically, it tells us whether a particular tryptophan, whether the average tryptophan on the molecule is exposed to water or whether it's hidden away from it. And so I intend to use this tryptophan located close to the core. Initially, it would probably be sequestered away from the water, but upon oxidation, I imagine that if the domain opens up, it would become more exposed to water. And so what this would look like is essentially we would start with this curve up here, where the fluorescence of the tryptophan residue is very strong, but upon being exposed to water, it would be quenched and become weaker. And this is generally what I observe, with a little bit of noisiness. So I find that as I increase the concentration of bleach, I see a general decrease in the fluorescence. There's a little bit of noisiness that characterizes this data, but that's probably due to the fact that there are other tryptophan residues on the surface of the protein. And I've observed before that this domain can dimerize, can kind of like stick to other instances of the CCB domain, which could potentially like sequester other residues and introduce some noisiness into the data. And so I fit that to a binding curve to see how sensitive the domain was to experiencing a structural change upon oxidation. And I found that at a concentration of about 200 micromolar bleach, half of the protein has undergone this structural change. And this is interesting because this is on about the same order as the cysteine being oxidized. So that indicates that the cysteine is readily oxidized, it's specifically oxidized, and this oxidation is correlated with a structural change that I can observe empirically. And so essentially, this demonstrates that this is quite possibly the mechanism that underlies bleach sensing, and that the structural change that ensues involves some kind of opening up of the helix. And so for further experiments, I'd be interested in doing more precise structural studies where I try to follow the movement of individual residues to try and get a better understanding of how the signal is transduced to the rest of the protein. And I'd also like to consider the cellular environment and how that might affect signaling, because I did most of these experiments with samples of purified protein, but bacterial cells are kind of this like bag of reductive systems. So it's quite possible that the experience that this protein has within a cell is very different from the experience it has purified and in solution. And also, I would also like to get a better understanding of the way CCB domains could vary amongst different bacterial species. And so I'd like to thank the Mary G. Alden Award and the Knight Campus Scholarship for supporting my work. And I'd also like to give special thanks to my mentor, Arden Perkins, and Dr. Karen Gilman and Dr. Jim Remington for advice and input.
Great, thank you. Yes. Anybody have questions for Dan? Okay, we've got a question here. During the bleach laser studies, is the gut sterile?
So the gut would not be sterile. it was a while since I read this paper, but my understanding is that this was within a conventionally reared mouse gut infected with H. pylori. So it has other bacteria in it as well, I think. So I think the idea was to have a true replicate of how the bacteria might interact within the mouse host. And I guess the caveat that introduces is that there could be some other interaction that is present. But we also know, but however, these trials over here where we observe that the bacteria is attracted towards bleach when it's dispensed from a pipette was done sterilely with just the pylori present in the camera's field.
Good. Other questions? Well, we have about 10 minutes left. So I would love to have a broader discussion here. We can come back to a few of the questions that we didn't get to, or people can raise questions that they didn't get a chance to ask, but now are coming to mind. I wonder if we can also promote anybody that wants to be promoted and have a discussion with them without just the chat feature, but them actually asking. Some of the mentors are still here. Anybody have a preference? Okay, here we've got a question. Actually, do you want to just read it, Alina? This is for you. Might be easiest.
Yes, thank you. Okay, so I think the question is if error-prone DNA repair pathways are suppressed by SMC56, NSE2, and BRCA1, and can rescue the progeny viability in response to environmental DNA damage, why would these potentially life-saving pathways be suppressed so strongly? And that's a great question. And I think really it comes down to not wanting to induce DNA damage or inducing further mutations because these, potentially mutagenic repair pathways, even if you do fix DNA and you introduce a mutation while you're doing this because you're utilizing a mutagenic prone pathway, then that one mutation essentially will end up becoming part of every single cell of the offspring's body because when the sex cells come together and they fuse, those are the chromosomes that you're left with that are then duplicated throughout your body. So I think that's why I hypothesize that they would be suppressed so strongly.
Other questions? Anybody? Okay, I'm going to jump in and ask a bigger question. And obviously we can't totally get at this in 9 minutes, but I'm curious how all of you are making sense of the sort of current moment we're in with coronavirus and COVID and sort of thinking about your own research. Obviously it's disruptive to your experiments and things like that, but is it making you think about where you want to go or the things that you want to do specifically with your research? Or more than anything, is it just making you want to get back in the lab and continue doing the research that you've been working on? Anybody?
I guess kind of like the observation that I keep coming to is just how this kind of viral infection strategy really takes advantage of the human need to socialize and be around people. And so it kind of gives me a better appreciation for the interaction between humans and virus and bacteria and how we're very much in sort of an evolutionary arms race with these microbes. And so I guess it does underscore to me the importance that my research has and the very real value that work like this has for the world.
Cool.
On the more lab side of things, I have found that there are a lot of things that I can do from home for research, like analyzing images. However, it also really exacerbates the things that I'm not able to do that without the lab and without the different, utensils for doing that.
Right.
So that's been an interesting discovery.
Right.
Not to be pessimistic, but it definitely ruined a lot of my project because I'm a senior, so I'm graduating in June, and I was supposed to get like a good amount of my data during my spring term because it just takes a long time to get a lot of data, but that's not going to happen because of the coronavirus. Like I'm going to leave, I'm leaving Eugene to go to medical school, so it's just kind of
Done.
yeah, it's done, and then you can't like go back, so it's
Well,
unfortunate, but
well, and it seems like, you'd encountered so many issues that you had to troubleshoot, then it slows it, and then when you're finally ready.
Yeah, exactly. So it was just like, well, so what was all this for? But it's okay. I still got the experience to like work in the lab and everything,
Right.
which is like helpful to just have that way of thinking. But it's just unfortunate that I didn't get what I was what I was hoping to get when I started the project.
Right.
I think for me, it's made me definitely excited to go back to the lab. I think when you're troubleshooting a lot, you can get burnt out pretty easily on research and lab work. And looking ahead, a lot of people who are on fellowships and grants are supposed to be doing full-time research for the summer. So, hoping that does happen. This was almost a nice little breather right before that starts, but maybe far too long of a breather because it's been a while.
Yeah, and I feel very similar to how Cailan feels. I think that I definitely do miss being in lab and just having this sort of hands-on approach. Although I have felt like I've picked up a couple more, tech-savvy techniques now that I'm at home, but there's definitely something about being in lab and being able to work hands-on, and also just having sort of that, like in-person collaborative nature of being in a lab that I definitely do miss. So I'm excited to get back.
Yeah, that lab environment is really hard to replicate online. And I love Zoom and I love all these other tools, but that face-to-face interaction and I think the stuff that comes out of, people being around and you're trying to troubleshoot something and you have somebody weigh in on something. I mean, just so much comes out of those kind of unexpected interactions. Any other thoughts that people have? We've got a question here, the olfactory study. Could it give insight into the early symptoms of lack of smell in COVID-19 at some point in the probably distant future? You're muted.
Sorry. Yeah, I was gonna say that it's possible, but right now I don't really see how it could relate to COVID-19, but I don't really want to say no, so I'll just say that it's possible. Yeah.
Other questions? Lots of congratulations coming in on the chat.
And I actually didn't know that like one symptom of COVID-19 was a lack of smell. I hadn't heard about that, so that's interesting.
Yeah. I don't remember what the percentage of patients that are experiencing that, but I believe it's well below 50%, but it's a sizable number of people. Before I end this, I will make one little plug in that a group of us is starting to organize contact tracing efforts related to COVID for Lane County. So within the next week, hopefully all of you will see a recruitment flyer. We'll probably have about 50 students. These can be current students or students who are graduating next month. But basically it's 3 months working with us and with the county and with the state on contact tracing of people who are infected. And so it's pretty exciting to bring in students to be able to do that. So hopefully all of you will see that, and hopefully all of you will be interested in pursuing that. So I guess with that, I will call the end of this a few minutes early and thank all of you for participating in this. The level of the talks was absolutely wonderful. The research that all of you are doing is just absolutely amazing. So thank you for presenting here. Thank you to all the mentors. Thank you to the mentors who showed up here and asked questions and supported your students, and best of luck to all of you moving forward.
Thank you.
Thank you.
Thank you.
Thank you.
Thank you.
And it's just us. Nice. I think that went well. And we can stop our recording right now.